Hello, and welcome to BioWorld. Today we will take a closer look at enzymes, which are essential in almost every biological process there is. They're vital in sustaining life on planet Earth. So first of all, what is an enzyme? To put it simply, an enzyme is a substance, most often a protein, that catalyzes a reaction. Well, what does catalyze mean? Catalyze is a very important word in many biological and chemical contexts, so it is a word that is crucial to your vocabulary. Catalyze essentially means speeds up. In this context, an enzyme's function is to speed up a reaction. A reaction can take 10 hours to complete as shown here. However, when there is a catalyst or enzyme involved, this same reaction can take a mere 10 seconds. Well, why is speeding up a reaction important? Chemical processes needed to sustain life need to be completed quickly, or else many of the normal processes in our body would not even work. Therefore, enzymes are necessary for life on Earth. Some processes that enzymes play a vital role in are the nervous system, which relays information between the brain and the rest of the body, the fight or flight response, the reaction the body has to outside stress, and the digestive system, which breaks down food in our body. All of these processes need to be done quickly in order for survival. Here is a drawing of an enzyme. An enzyme, as mentioned before, is a protein, and its structure and therefore function depends on its amino acid configuration. Each enzyme has different properties based on what amino acids are part of its structure. This is the active site, where the substrate, which will be talked about later, will bind to. This is the allosteric site, which is far from the active site and sometimes present, but plays a big role that will be discussed later. The basic overview of what an enzyme physically does is as follows. The substrate, usually the reactants of the reaction, binds to the enzyme at the active site. The shape of the substrate is always complementary to the shape of the enzyme's active site. Then, to cause a tighter hold on the substrate, the enzyme pretty much hugs the substrate, causing what is called a conformational fit. The reaction then proceeds and the products are released. Enzymes work best in certain environmental conditions, which are called optimal conditions. Each type of enzyme has its own optimal conditions in temperature, pH level, salinity, etc. These depend on the amino acid configuration of the enzyme. In the graphs below, the point at which the rate of reaction is highest is the optimal condition. However, different enzymes have different optimal conditions, as shown by the orange dataset. For example, stomach enzymes that help in digestion are at their optimal condition at low pH levels, due to the fact that the stomach area is very acidic. On the other hand, enzymes in the blood may have optimal conditions at neutral pH levels, because blood has a very neutral pH. However, why do enzymes work in certain conditions and not in others? In this example, the enzyme is intact and working at temperatures below 40 degrees Celsius. However, it breaks and changes shape at temperatures higher than 40 degrees. This is because the heat denatured the enzyme or broke it apart. The excess heat broke many bonds holding the enzyme together, eventually leading it to change shape and not be effective anymore. This concept is present in other circumstances like pH and salinity, as excess of both can break bonds in enzymes too. However, at very cool temperatures, the rate of the reaction is still low because of the fact that the molecules, which include both the enzyme and substrate, are not moving enough to cause many collisions with each other, causing a lower rate of reaction. Now, we will return back to the allosteric site. This site is away from the active site, and complementary molecules can bind there. However, these molecules can be used to either activate or deactivate the enzyme by changing the shape of the active site upon binding, as shown here. The allosteric inhibitor binds to the allosteric site and changes the shape of the active site, causing the substrate not to be able to bind with the active site and deactivating the enzyme. Well, why is this needed? The body needs to regulate the amount of certain substances, and too much of something can be fatal. In this example, the first enzyme produces a product that is the substrate of the second. Then, the second enzyme does the same for the third enzyme. However, the third enzyme then produces an allosteric inhibitor for the first enzyme, which stops the whole process from occurring. This is called a negative feedback loop, which stops excess product from forming. Positive feedback loops increase production exponentially by using an allosteric activator rather than an allosteric inhibitor. Thank you for watching. If you enjoyed the video, please be sure to leave a like and subscribe for more bio videos.